welcome to the second video unit of the introductory module to seed quality and health, presented in the frame of the Live Seeding project. As presented in the first video unit, this module can be taken as a standalone general introduction or as part of more in-depth training paths offered on the project website. My name is Stéphanie Kletke, and I work at ITAP, the French Organic Food and Farming Institute. Based on what I presented in the first video unit, in this one we will explore factors that drive seed and plant health. We will then have a closer look at interactions between seed vigor and health. To finish, I will present some critical situations for seed health and quality in organic systems as examples to understand the scope. To begin with, let's get an overview of the many factors that affect seed health and quality. To do so, I'll go back to the scheme presented at the end of the first video unit. Remember, seed and plant health depend on each other in an iterative process, and that seed vigor has major importance to ensure well-performing organic crops. To start with, when propagating seed, it's obviously important to choose cultivars or varieties adapted to your context. Weather and soil conditions will also influence the quality of the seed produced. Throughout the cultivation process, you need to keep a close eye on the plants to ensure they are in good health and, if necessary, eliminate diseased plants or select for the best seed-bearing plants. Harvesting conditions are of utmost importance, and care must be taken to ensure that seeds are harvested with appropriate techniques at full maturity. Once harvested, the seeds must be dried thoroughly to preserve their physiological quality and maximize their storability. Seed sorting and cleaning contribute to seed quality by discarding plant debris, weed seeds, and any seeds that are too small or lack maturity. Testing seeds for their germination and maybe also vigor throughout storage allows you to monitor seed conservation and the quality of the seed that is distributed. According to crop species and context, testing seeds for the absence or presence of seed-transmitted pathogens makes sense as a routine or if a disease was suspected in the seed crop. Sometimes treatments are required to sanitize the seeds. These can be routine preventive treatments, as for example recommended against common bunt in wheat, or they can be performed after seed analysis has indicated the presence of a pathogen. These aspects are addressed by Jelena Bacanovic-Sisic in Module 15 on hot water seed treatments. All this has an influence not only on germination capacity, that is the germination rate of the seeds, but also on their vigor and resilience. Research and organic seed production practice still have a lot of work ahead of them to understand how seed microbiota can best be taken into account in the management of seed-transmitted plant pathogens. The conditions at sowing, and especially soil conditions, have a strong impact on the emergence and health of seeds too, of course, as the seedling is in close interaction with the soil during the germination process, both from a physical and a biological point of view. However, sowing environment and conditions are not something you can control as a seed company or farmer seed network or community seed bank when you distribute seed. Maybe you've noticed that I often use the term seed quality and health and don't really make a clear distinction between seed quality on one hand and seed health on the other. Although seed quality usually refers to physiological aspects and seed health to microbiological aspects and absence of pathogens, these two domains actually interact with each other and are not completely separated. I would like to give you two examples to illustrate this. The first example is from an experiment that was conducted by Steven Groth at Wageningen University and Research during the Live Seed project to study the effect of seed vigor on Alternaria radicina, which causes damping off of carrot seedlings. Seeds were sprouted in petri dishes and inoculated with the fungal pathogen. In the petri dish on the left, you see the sprouted high vigor seed lot. On the right, you see the same seed lot which had previously been artificially aged to obtain the same seed lot but with a lower vigor. What you can see is that high vigor seed results in healthy seedlings, while seedlings with the low vigor on the right damp off under the disease pressure. This illustrates that physiological seed quality contributes to seed health by making seedlings more resilient to biotic stress factors such as soil-borne diseases. With another example, I want to show how seed physiology affects the management of seed-borne diseases through seed treatments. In France and Denmark, vinegar treatments at about 4% acetic acid are frequently used as preventive disinfection against common bunt in organic wheat seeds. In a lab experiment in collaboration with ITAB, the French organic seed cooperative Ubios tested the effect of seed treatment of different wheat seed lots at different vinegar concentrations, up to 10% acetic acid. In one treatment shown in light blue, slaked lime was added to neutralize the effect of the high concentration vinegar. What I want to show you here is that some seed lots are more resilient to the seed treatment than others. In the graphs, you can see the germination curves of two seed lots according to seed treatment. Seed lot A on the top had much lower vigor. You can see that even the non-treated sample in dark blue sprouted only slowly and had a maximum germination rate of about 78%. You can also see that some of the seed treatments had a strong negative impact on the germination curve, especially the vinegar treatments at 8 and 10%, in green, brown, and light blue. In the graph, germination is even slower and final germination rates are even lower than the non-treated version. Seed lot B on the bottom had much better physiological seed quality, as the seeds sprouted much more quickly and homogeneously to reach a maximum germination rate above 90% when untreated. Here, the vinegar treatments at 10% in brown and light blue seem to have affected the germination curve a bit, but final germination rates remain almost stable. This example illustrates that caring for high physiological seed quality also widens your opportunities to treat your seeds against seed-borne pathogens, if necessary. Now that we have seen that microbiological and physiological aspects of seed quality are intertwined, I will describe some typical critical situations in organic seed systems to understand the importance and scope of managing for seed quality and health. In a plant breeding process, it is crucial to avoid breeding material getting infected with seed-borne diseases, as this may disqualify promising elite lines or populations in the breeding process and make propagation of new cultivars difficult from the start. For example, in wheat again, it would be problematic to have common bunt caused by fungi of the genus Tilletia in your breeding nursery. The good news is that the small plots usually used at the beginning of a breeding process are much easier to monitor for plant health than large production plots. Roguing early on, that is removing any plants with disease symptoms to lower the risk of infecting other plants, is thus easier in this context. In some cases, specific sanitation practice can also be applied to small breeder seed lots. In our common bunt example, you could wash seeds of small seed batches with water or apply a hot water treatment, which is not easily feasible with very large seed lots. Another noteworthy type of situation is when you want to improve a population's tolerance to a given stress based on so-called natural selection by applying the stress to the population. For example, some farmers prefer to make on-farm seed production under conditions of hydric stress in order to favor drought tolerance in their maize or tomato crop. This may certainly work from a selection point of view to improve the population genetically. However, also have in mind that seed crops that have some kind of nutrient or water deficiency will often produce seeds that are less well filled or mature too quickly. This may result in lower seed vigor and storability of the seeds. In such cases, it is preferable to dissociate the application of a selective pressure and the seed propagation in separate steps. You could, for example, harvest seed after selection under hydric stress or low nutrient conditions as your base or pre-base seed to further propagate the population, which was genetically improved for stress tolerance. The seed propagation of the improved population could then happen under conditions which are as optimal as possible for plant health and nutrition, in order to produce high-quality seeds for distribution. I just want to stress the importance of distributing high-quality organic seed for seed companies, seed networks, and community seed banks in the interest of the people using their seeds and, in the end, in their own interest. Many organic seed companies and networks do a great job in offering seed of population varieties adapted to local organic growing conditions. A lot of effort is put into choosing, evaluating, sometimes improving these varieties from a genetic point of view in order to distribute best adapted and diverse varieties and cultivars. It is really important to put a lot of attention and care into seed propagation, harvest, storage, and handling to ensure best possible seed quality when distributing these varieties and cultivars. Indeed, seed quality affects the performance and health of crops of seed users, that is, market gardeners and farmers. As they are the ones incurring economic consequences of poor seed quality, it is often enough for them to have 1 or 2 bad experiences with the quality of seed obtained from a seed company or network to stop taking seed from them. In this way, a seed company or seed network, or even population varieties in general, can easily be discredited by farmers and gardeners based on just 1 or 2 bad experiences with seed quality and health. To recap on this video unit, I would like to remind you that seed quality and health are intertwined and that seed management requires complementary strategies. On the one hand, seed should be strengthened by optimizing and preserving seed vigor throughout the production cycle. The aim in future is also to preserve healthy and beneficial seed microbiota in a holistic approach, but the functional understanding we currently have of seed microbiota is not yet advanced enough to really allow for this. On the other hand, even if we strengthen seed as much as possible, it is also necessary to protect seeds from plant diseases, especially by monitoring diseases and potential pathogens in seed crops and seed lots, and sometimes through seed sanitation. Of course, the content presented in this video is only a quick glimpse at the necessary theory and background knowledge to manage seed quality and health in organic seed systems. In practice, it requires experience with local conditions, the respective crop and cultivar, and predominant stress factors and plant pathogens. Thank you for your attention. I hope you enjoyed and learned from the second video unit. To take things a bit further and finalize the training module, I suggest you look into the reading material 11.3 and then take the quiz 11.4.